The broadcast is now starting. All attendees are in listen-only mode.
Hello, everyone. My name is Iris. I'm from GenScript Marketing Department. It is really my pleasure to invite Dr. Liu Zongying to talk about bispecific antibody, the next generation therapeutics. Dr. Liu Zongying is a director and head of the antibody department at GenScript, responsible for antibody drug discovery, monoclonal and polyclonal antibody generation, and immunoassay development. Dr. Ying received his PhD degree from University of Massachusetts Medical School, specialized in antigen presentation, CD4 T-cell epitope selection, and antibody responses. He then joined Pfizer, specialized in immunogenicity of therapeutic biologics. Dr. Ying has extensive experience in antibody drug discovery, from lead generation to lead optimization, especially in DNA immunization, high-throughput FACS screening, ELISA kit development, and anti-EDODAP antibody development for antibody drug PK/PD studies, as well antibody humanization, affinity maturation, and developability assessment. Dr. Ying has published over 14 well-cited papers in major immunology, variety, and biochemistry journals, and 10+ patents in antibody discovery field. In this webinar, first off, we will cover an overview of therapeutic antibodies, focusing on the opportunities and challenges of current monotherapy. Next, we will discuss the major benefits of bispecific antibodies and the current formats used in clinical trials. The last part will be an in-depth integrated SMAB case study for rational design to preclinical development. To ensure the sound quality, all the attendees are in listening-only mode. Please feel free to type in your questions at the chat box in the lower right corner of your screen throughout the talk. Dr. Ying will address your questions after the presentation. Without further ado, let's welcome Dr. Ying to give his talk on bispecific antibody, the next generation therapeutics.
Thank you, Iris, for the introduction, and thanks everyone for attending this webinar. As mentioned, I will talk about bispecific antibody, the next generation therapeutics. So as GenScript is a public company, this is a disclaimer and a forward-looking statement that this presentation is for information purpose only. So this is the outline of my presentation. I will first give an overview on therapeutic antibodies and focus on the opportunities and challenges of current monotherapy. Aiming at addressing unmet medical needs, we will then discuss major benefits of bispecific antibodies and review current platforms for that. I will also introduce GenScript's proprietary SMAB bispecific antibody platform, which minimizes the immunogenicity and the manufacture concerns of current bispecific antibody platforms, while enabling bivalent and multivalent therapeutics. Lastly, we will give a case study for SMAB bispecific antibodies, from rational design to preclinical development. At the end of my talk, I will give you some facts about GenScript, and I'm happy to answer your questions after that. So I will first give an overview of antibody therapeutics. Therapeutic antibody drugs have recently experienced explosive growth. Within oncology research alone, more than 20 therapeutic antibody drugs received FDA approval for new treatments or indications during 2015 to 2017. Additionally, more than 300 therapeutic antibody drugs are in ongoing clinical trials. Most recently, the success of cancer immunotherapy by blocking immune checkpoint proteins such as PD1 and CTLA-4 created a hope for cancer cure and triggered a second revolution that the majority of pharmaceutical companies are devoting major research force and investment in antibody therapeutics. Therapeutic antibodies function through multiple mechanisms of actions to battle cancer, including direct inhibition or apoptosis of cancer cells, like this anti-HER2 antibody Herceptin for breast cancer, or through ADCC, antibody-dependent cytotoxicity, and CDC, complement-dependent cytotoxicity, like anti-CD20 antibody retoxin for lymphoma, or through immune cell activation and recruitment, like blockbuster anti-PD1 and anti-CTLA-4 antibodies for multiple cancer indications, and anti-CD19 first ever CAR T cell therapy for liquid tumor, or through cytotoxin delivery like ADC, antibody drug conjugates. So let's take a look at 2017 FDA newly approved drugs with novel first-in-class mechanisms. As you can see, most of them are biologic therapeutics. And among them, the majority are antibody-based therapies, including the first anti-interleukin-2 and interleukin-4, the receptor duplexin for the treatment of allergic disease, and the third bispecific antibody, Hybridoma, for the treatment of hemophilia A, as well as the first CAR T cell therapy, Chimera, for the treatment of B-cell acute lymphoblastic leukemia. To summarize, therapeutic monoclonal antibodies have made many breakthroughs and brought many meaningful treatments to patients and physicians. However, even though compared with conventional chemotherapy, monotherapy, especially in immuno-oncology field, has significantly increased the survival rate. It still faces many unmet medical needs, in which combination therapies and bispecific antibodies can offer particular advantage. So as you can see in a BMS Checkmate 0767 phase 3 study in advanced melanoma, combination of anti-CTLA-4 Yervoy and anti-PD1 Opdivo showed much improved median progression-free survival compared with Yervoy or Opdivo monotherapy. However, this enhanced efficacy was associated with increased grade 3 to 4 adverse effects, from less than 30% to almost 60%. So as mentioned, both bispecific antibodies and combination therapy can address many unmet medical needs. However, bispecific antibodies can offer additional advantage. Firstly, bispecific antibodies can provide superior potency due to novel mechanism of action. For example, the first generation bispecific antibodies are almost T-cell engagers, in which the underlying principle is quite straightforward, with bringing T-cells to target tumor cells. Other novel MOA include moderation of receptor signaling and simultaneously targeting of multiple core inhibitory receptors or checkpoints, like bispecific antibodies against both HER2 and HER3, and against PD1 and CTLA-4, as well as the third approved bispecific antibody, Hybridoma, targeting factor 9 and factor 10 simultaneously for the treatment of hemophilia A. Bispecific antibodies can also target multiple epitopes on the same target for synergistic effect. Secondly, bispecific antibodies can offer enhanced safety profile due to less off-target binding and presumably lower dose, as we can see safety concerns in combination therapies. Thirdly, developing bispecific antibodies is more cost-effective as it only develops one molecule and saves half of the investments in comparison with combination therapy. All these advantages of bispecific antibodies make them a potential powerful treatment for patients and physicians. So to date, there are only 3 approved bispecific antibodies in the market, but there are hundreds in clinical trials. With the novel MOA, enhanced safety profile, and a controlled price, the bispecific antibody market size is estimated at 450 million in 2018 and will reach 20,000 in 5 years. So this is a short list of in-clinical bispecific antibody strategy by many world-leading biotech companies and biopharmas, with multiple mechanisms of actions, including T-cell, NK-cell recruiters, to ligands inactivation, to factor dimerization, targeting to immune checkpoints, and to break the blood-brain barrier or induce more internalization. So in the next few slides, I will talk about various bispecific antibody platforms and focus on GenScript's SMAB bispecific antibody platform. So this is a schematic overview of the different strategies used to generate bispecific antibodies derived from the antigen binding sites of two different antibodies. Symmetric bispecific antibodies are generated by the assembly of antibodies with unmodified heavy chain constituent regions, such as by heterodimerization of heavy chains from two different antibodies or homodimerization of heavy chains extended by an additional binding site, resulting in bivalent or tetravalent molecules. Using heavy chains modified to force heterodimerization, for example, using a Knobs-into-Hall strategy, results in asymmetric bispecific antibody. Alternatively, two different antibody fragments such as SFA can be used to a non-immunoglobulin protein such as albumin. Furthermore, two antigen binding fragments can be directly fused, resulting in small bispecific antibody molecules. Finally, bispecific antibody can also be generated by chemical conjugation of two different antibodies. To date, there are about 200 different bispecific formats, and the most common 20 formats have generated various candidates in clinicals or even approved by FDA and EMA. So despite the numerous bispecific antibody formats and the benefits that bispecific antibody have brought to patients, there are two major concerns for bispecific antibodies. Firstly, as most of the current formats are over-engineered, and therefore they may have immunogenicity concerns, as our immune system is very sensitive to unnatural stimuli. Secondly, as these bispecific antibodies often have unnatural formats, they are not stem cell friendly or have manufacturer concerns, including product instability, lower expression level, and the complex purification process. So with these two major concerns in mind, we have developed GenScript SMAB bispecific format, which is a single domain antibody fused to a monoclonal antibody. Our concept is very simple, that we will use all nature components to realize bivalent and multivalent purpose. Therefore, in our SMAB format, we use nature single domain antibodies fused to nature monoclonal antibodies with nature or less immunogenic linkers. In this setup, SMAB has symmetric structure without FC engineering, which brings many advantages. It has the same single-step protein A purification process. And since it has all nature antibody formats, the durability of SMAB antibodies are comparable to conventional monoclonal antibodies with high yield and ability for high concentration formulation. Moreover, since we use single domain antibody as part of the binding partner, SMAB is very flexible with ability to be multivalent. So this is a direct comparison of SMAB with other bispecific formats, including Zymeworks asymmetric bispecific, Abelin's nanobody, GenMab dualbody, and AbbVie DVD-Ig. Zymeworks asymmetric format with FC engineering may have immunogenicity issues, which will be tested in several clinical trials. Abelin's nanobody format faces the issue of short half-life, and they are developing several strategies to overcome the problem. GenMab's dualbody is involving post-production processing to generate the bispecific antibodies. AbbVie's DVD-Ig may introduce steric hindrance due to rigid conjugation of two antigen binding sites. So the SMAB format uses a symmetric structure favoring production and stability, while preserving long half-life and minimize immunogenicity, but we still have to test this in clinicals. So in SMAB format, we use single domain antibody as one part of the binding partner. The reason is that single domain antibody has many advantages. The size of single domain is small, so it can bind to the epitope normally hidden from conventional IgG, such as cavity of enzyme, and it has better tissue penetration ability. Because the single domain antibody has only one domain, it is easier to engineer to other formats. So compared to conventional IgG, most single domain antibodies are more stable and can tolerate high pH and temperature. that enable the antibody to be delivered via multiple routes instead of IV only. Moreover, the flexibility of single domain makes it suitable to various therapeutic purposes. Due to its small size and easy to engineer, bi or multi-specific single domain antibody can be formed to target multiple targets or multiple epitopes on the same molecule at the same time to increase therapeutic potential. So for single domain antibody generation, we can use library approach, either naive library or synthetic library or immune library. Here I show a general workflow for single domain discovery using immunized llama or camel. After preparing immunogens and the immunization of llama and camel, phage library will be constructed and screened. We then use the patent FASIBA high-throughput platform to screen for binders with good expression, affinity, and some stability for downstream further characterization. For MAB generation, there are several approaches. Over the 70 therapeutic antibodies approved by FDA and the EMA, the majority, over 60, were generated by hybridoma approach, including hybridoma with transgenic animals for fully human antibodies, and rodent hybridoma with humanization optimization. The rest 8 were generated through phage display library approach. Other approaches are also available for MAB generation, including single B-cell based approach coupled with next generation antibody sequencing. So this is a comprehensive case study for MAB lead generation using hybridoma approach. In this practice, 10 mice were immunized and the test bleed were evaluated by both ELISA and FACS. After that, 2 animals with top titers were performed for fusion, and the generated hybridomas were plated into 65 96 well plates. So after primary and cumulative screening, over 800 ELISA positive wells of parental clones were identified, and 86 of them were confirmed with ligand blocking activity by ELISA, of which 26 parental clones were confirmed with binding by FACS, and they proceed to one-round subcloning. 53 one-round subclones were obtained, and we selected 28 of them for antibody microproduction. Those 28 antibodies were validated by mixed lymphocyte reaction functional assay, which is a standard function assay for immune checkpoint antibody lead identification. So after the single dose MLR functional assay, 12 of the one-round subclones were further subcloned to obtain stabilized functional clones, and then larger scale antibody production was performed. For those 12 antibodies, multiple primary hits characterizations, including ELISA EC50 and FACS for binding activity, ELISA IC50 and FACS for ligand blocking activity. So after those characterizations, 5 antibodies were selected for final leads characterization, including those multiple dose MLR function assay, epitope binding, cross-reactivity, affinity measurement, and sequencing. In this way, we showed how functional leads were identified through hybridoma approach. So this slide shows another approach, the naive library to generate fully human antibodies for bispecific construction. In this approach, PBMC were isolated from healthy donors, and the antibody heavy chain and light chain variable domains are amplified and constructed to Fab library for screening. The key parameters for naive library is diversity and library size to ensure the diversity. Human naive library can generate fully human antibodies, but may have undesired affinity. In that case, affinity maturation may be required to further optimize the leads. So each lead generation platform has its own advantage and disadvantage. So a quick comparison across different platforms, combining quality, cost, and turnaround time. Hybridoma approach has a major advantage of generating antibody leads with good durability, but may encounter immune tolerance issue or has to be immunized from rodent hybridomas if not working with expensive transgenic animals. Phage display approach can screen out numerous hits within a relatively short time frame, but may encounter poor durability. So single B-cell approach is able to generate fully human antibody sequence but usually limited to infectious disease. So most often, many approaches are utilized in parallel to maximize the success rate of lead generation. So in the next few slides, I will talk about antibody lead optimization for bispecific antibody generation. So before a therapeutic antibody lead entering downstream CMC development process, 4 lead optimization are often performed to ensure the druggability or minimize the downstream development risk, including antibody humanization, affinity maturation, durability assessments, and immunogenicity evaluation. So due to high immunogenicity of murine and chimeric antibodies, humanization is required for rodent leads. Antibody humanization is a process of replacing non-human antibody frameworks with human ones. Successful antibody humanization depends on maintaining the affinity after replacing residues. In general, there are 2 kinds of humanization strategy: CDR grafting, which does not change human germline, and germline shuffling, which change human germline. Here I show a general workflow for antibody humanization. So antibody sequence is analyzed to select best human germline acceptors for marine CDRs. Post-translational modifications are checked to avoid poor developability of selected germlines. So after CDR grafting, homology structural modeling can be performed to identify back mutation residues and use high-throughput screening to identify antibodies with good affinity, expression level, and thermal stability. So here I show an example of humanization of an anti-cytokine single-domain antibody. So we selected germline with high sequence homology, and after homology structural modeling, we identified 16 framework residues that were different, highlighted in red, of which 5 positions were modeled to be potentially critical for antigen binding close to CDRs and the putative back mutations performed. So after this humanization process, we successfully obtained a humanized single-domain antibody with comparable affinity and functions. Affinity maturation is another lead optimization process, which is aimed to increase affinity of selected antibody leads. Usually, leads from hybridoma approach have reasonably high affinity with KD in nanomolar to sub-nanomolar range, and therefore antibody affinity maturation may not be necessary, especially with affinity guaranteed humanization process. However, in some cases, that the obtained leads have relatively low affinity, such as from phage library campaign and transgenic animals, or the final application needs extremely high affinity, affinity maturation is required. Here we show the flowchart of the affinity maturation process. After the affinity antibody sequence is obtained and confirmed with its binding to antigen, we will perform the parietal mapping to identify the key residues for antibody antigen interaction. An NNK single point mutation library is built and screened to select top binders. Then combinatorial library is built and screened to select best binders. So the affinity matured IgG is produced and characterized. After this affinity maturation process, the affinity usually will be increased by at least 5 to 10-fold, and we have seen cases of increasing 40-fold. So for therapeutic lead selection and optimization, antibody drug developability assessment is very critical. We mainly focus on chemical stability, physical stability, and solution properties. One key developability risk is post-translational modifications such as asparagine and glutamine deamidation, aspartate aspiration, methionine and tryptophan oxidation, free cysteine, N&O glycylation, and paraglutamate lysine glycation and isoelectric point cost aggregation. So various tools are utilized to assess durability risk, including in silico structural modeling, size exclusion chromatography, peptide mapping by LC-MS, and affinity by SPR-based vehicle. So for developability assessments, we can use these methods for biophysical characterizations, including SAC HPLC, ELISA, DSC to check thermal stability, aggregation, free source stability, biometric stability, expression level, and solubility properties. For post-translational modification hotspots identification, like asparagine deamidation, aspirate aspiration, we can use peptide mapping, followed by LC-MS and check binding by ELISA of Buccal. In case there are developability risks identified by these methods, antibody engineering will be further implemented to remove or mitigate the risk. So immunogenicity of therapeutic antibodies is caused by various product-related and patient-related risk factors. We can cause different therapeutic outcomes in terms of drug efficacy and patient safety. Various methods have been established for measuring and predicting immunogenicity, and FDA has guidelines for risk-based mitigation efforts for immunogenicity. There are several layers of immunogenicity risk assessment, including in silico prediction of T-cell epitopes and B-cell epitopes. Then, multiple in vitro and ex vivo assays will be developed to evaluate the in silico prediction, including DCMS assay, PBMC assay, and whole blood assays. Lastly, non-human primates will be used to test the immunogenicity of therapeutic bispecific antibodies in vivo. And the endotype antibodies are often developed to be used as reference standards or positive control in immunogenicity assessments. So here is a quick summary. We have talked about various strategies to construct bispecific antibody, including symmetric and asymmetric formats, different bispecific antibody conjugation methods with or without Fc fragments, nature or Fc engineering. The concept of GenScript SMAB bispecific platform is to be nature with good developability and safety profile. So during the bispecific antibody discovery, antibody lead optimization is a combination of bioinformatics, antibody engineering, and high-throughput screening and evaluation tools to make sure the bispecific leads we generate are fit for downstream development process for clinical usage. So in the next few slides, I will give you an example of SMAB bispecific antibody from rational design to preclinical development. So we select PD1 and CTLA-4 as the bispecific targets. As you all will know that PD1 and CTLA-4 stand at the center of the breakthrough immuno-oncology axis. Even though the detailed function mechanism of PD1 and CTLA-4 antibodies are still in debate, anti-CTLA-4 antibodies are thought to block the inhibitory signal between antigen presenting cell and the T cell during primary phase of T cell activation, while anti-PD1 antibodies release the negative regulation between effector T cell and cancer cell, both of which can enhance T cell effector activation. So besides that, there are numerous PD1 and CTLA-4 combination therapies, and also there are numerous PD1 and CTLA-4 specific antibody formats in other formats in clinical trials. So that's why we select the PD1 and CTLA-4 as our case study. So this is the first case study that we show that we construct a PC101 SMAB molecule comprising the monoclonal antibody anti-PD1 and the single domain CTLA-4 antibody. As you can see that both the SMAB antibody, they show very high affinity to this PD1 and CTLA-4 molecules. And from this design profiling phase, so we have evaluated an affinity of and also expression level of different bispecific format. So including the antibodies linked, the single antibody links at the N terminal or C terminal, or the light chain and heavy chain, and it also use different linkers. So after this design and profiling process, we have selected antibodies with good expression, with good affinity, and also some stability properties. And this is in vitro property summary of this PC101 molecule. So we checked the affinity to both PD1 and CTLA-4. And we also checked the cell-based binding to cell lines overexpressing PD1 and CTLA-4. We also checked the ligand binding competition with PD1 and CTLA-4 ligands, and checked the in vitro function assays. As you can see, this bispecific antibody PCA101 showed a very good affinity, ligand block activity, and functional assay to both PD1 and CTLA-4 molecules, and is comparable with those single molecules like the PD1 antibody and the single CTLA-4 single domain antibody. So next, we check the durability, because as you know that durability is key, that whether an antibody can enter the downstream development process. So for this PCA101 molecule, we check numerous durability parameters, including the transient expression, purity, thermostability, aggregation, and solubility, as well as cross-reactivity and non-specific binding and stability. As you can see from this summary slide, that they show very good durability with comparable expression level, purity, and stability. Then we select this molecule to go to the downstream in vivo activity test. So we use two different models. The first model we use is a synthetic model. And we directly engraft human PBFC into this mouse. And after we check this in vivo activity, we compare with both the monotherapy, Keytruda monotherapy, and the single domain monotherapy, as well as the combination therapy. So as you can see from this data, that the PCA101, the SMAB bispecific molecule, showed a very potent efficacy compared with the monotherapy, and is comparable and also a little bit better than this combination therapy. So we also use another model. We directly implement the MC38 cells into this PD1 and human CTLA-4 double knocking mice. In this model, we perform the similar in vivo efficacy study. So as you can see from this study, the conclusion is very consistent that this PCA101 molecule showed a better efficacy compared with this monotherapy and is comparable with the combination therapy. So just a quick summary of this PCA101. The PCA101 SMAB was determined to retain the in vitro potency of parental monoclonal and single domain antibodies. And the results of preliminary durability assessment supports PCA101 as a candidate for development. And the PCA101 was demonstrated with in vivo efficacy superior to monotherapy and equal or better efficacy compared with combo therapies. So just a quick summary of bispecific antibody. So bispecific antibody, as mentioned, they can meet many unmet medical needs. So what we expect in bispecific antibody, that we expect that they should have better response rates, more durable response, and better tolerability, or higher or better safety profile. And when you develop a bispecific antibody, especially using SMAB platform, we need to think about this, whether they have synergy of the target biology, and we have to think about this efficacy, durability, and safety of these different bispecific antibody formats. So just a quick summary of GenScript SMAB platform. So this is a bispecific bivalent or multivalent open access platform that we try to be nature. We try to develop bispecific antibodies with good durability, lower immunogenicity, and good efficacy. So in the last few slides, I will give you a quick overview about GenScript and our facilities. So GenScript is a world-leading biology CRO founded in 2002 in New Jersey, and currently is the #1 in the world for gene synthesis with over 2,000 employees worldwide. So GenScript is publicly traded in Hong Kong stock market in 2015 and specialized in antibody drug discovery and development. Here is a quick overview of the global presence of GenScript. So with the headquarters in New Jersey, USA, and main manufacturer and R&D centers in Nanjing, Zhenjiang, and Jinan, China. We also have branch office in Netherlands and Ireland, Europe, and Tokyo, Japan. So with the goal to serve worldwide researchers with highest efficacy and expertise. So this is the overview of GenScript platform for antibody drug discovery and development, from target to IND. lead generation and the lead identification is the start. So after getting the antibody leads from hybridoma or library approach, antibody engineering is performed for lead optimization, including antibody humanization, affinity maturation, developability, and immunogenicity assessment. After an optimized lead is obtained, we go to the downstream commercial step cell line development and the CMC process. So multiple testing modules are in place for biochemical, biophysical, in vitro, and in vivo characterizations, and further toxicity, stability, and durability studies, which are required for IND filing. So over the last decade, we have successfully delivered over 300 therapeutic lead generation and over 100 lead optimization projects, as well as 25 CMC projects. So more than 3 of them were moved forward to IND filing stage by end of 2017, with 1 approved for clinical trial. So this is just a quick summary of discovery platform. So we want to accelerate your drug discovery with innovation. So by acceleration, we have all this integrated platform. So we have used this power of integration and use the expertise and experience we accumulated over the 400 therapeutic antibody discovery projects delivered and with industrial standards. So we understand the standards of the molecule itself as well as documentation regulatory standards. And we use a large number of bioassays to support for identification of functional lead with good durability. We also accelerated discovery by innovative platforms, including this SMAB bispecific antibody platform that I just introduced, and also with the human naive library approach to identify fully human antibodies with better immunogenicity profile. We at GenScript value clients' confidentiality and intellectual property as highest priority. So we have a very well-established IP protection system, from sign of confidentiality agreements, secure online information transfer, IP and confidentiality trainings, compartmentation of clients' information to close the system with firewalls, password and email control and data backup. Notably, our IP protection system has been audited and approved by numerous world-known biotech and biopharmaceutical companies. So with that, we also have this one-stop solution from target to IND as just introduced. So we start from target validation and through this antibody lead generation and categorization. Then we further optimize the lead. And after that, we go to the downstream step cell line and the process development. And also we are going to this GMP manufacturing to directly move your from target to IND and from target to commercialization. So with that, I would like to thank you for your attention, and I hope you have learned something useful for your antibody projects. So I will pass the ball to Iris for Q&A session, and I'm more than happy to answer your questions. Yeah, thank you.
Okay, thank you for Dr. Ying's presentation, and thank you all for attending this webinar. Now it comes to the question and answer section. Let's take a look at the first question. Good morning, Dr. Ying. Do you see any advantage of combo therapy over bispecific antibodies?
Yes, sure. I think, as I mentioned, the combo therapy offers a lot of advantage. And the major advantage of combination therapy over bispecific antibodies is that the combo therapy is very flexible. So you can adjust the different dosing scheme of these different combinations, such as just a great example that for the first approved combo therapy, the Yervoy and the Opdivo. So they use a lower dose of Yervoy and use a higher dose of Opdivo. And so I think that's the major advantage of this combination therapy to adjust the dosing strategy. And another advantage is that because especially for this immune checkpoint targets, so for the combination therapy, and you can adjust the route. For example, you can first administer the PD1 therapy, then administer the other, like say OX40 therapy for better efficacy, because the immune system, they are not acting as simultaneously. Sometimes they have different, just like a different way for activation. Yes, thank you.
And the second question is, does SMAB have immunogenicity issue?
Yes, SMAB has a natural format, and its immunogenicity should be much lower than other asymmetric formats. But we are still evaluating the immunogenicity, and the common workflow is what I just described in the presentation, including the in silico prediction, in vitro, in vivo assays, and also the in vivo study. And we are building the prediction and of the evaluation platforms to evaluate the SMAB immunogenicity concern. But we have to see for sure that in clinicals.
And there's a third question for you. Is humanization needed in SMAB construction?
Yeah, I think we have several proprietary peptide linkers, not a chemical linker, to connect single domain antibody and one chain of molecular antibody. Yeah, but the linker is through covalent bond, like one chain. So peptide is not necessary for SMAB, and we can directly link single domain antibody with MAB in some cases. So with our linker, we will have a reduced immunogenicity concern and avoid the risk of interaction and digestion of this different sites of linkers. But we still suggest use linker to avoid the static hindrance to ensure the efficacy. So our peptide linker is a natural human sequence, and the risk of immunogenicity is very low.
Okay, due to time limitation, we will end up this webinar soon. And you can find relevant information through our website, www.genscript.com. And please do not hesitate to contact us if you have any questions, including but not limited to this webinar. Our technical manager and Dr. Ying will get back to you soon, within 24 hours. Thank you again for attending this webinar. I look forward to hearing from you soon. Have a nice day. Bye-bye.
Okay. Yeah, bye-bye. Thank you.